genetics. To be honest with you, in future medicine, your job in future definitely based on personalized medicine. What I mean, treatment for each person is different from other person. I mean, a special treatment for everybody. That's the results of personalized medicine. As an introduction for personalized medicine, I want to back to your high school. Your high school, you remember human body composed of cells, and each cell has got nucleus. In the nucleus is DNA, and then RNA in cytoplasm and protein production and function. You remember, as I said to you, that's from high school. Recently, in each part of the cell, DNA, protein, we have many X, such as metabolomics, genomics, transcriptomics, proteomics. Transcriptomics means RNAs. Transcriptomics RNA, genomics DNA, proteomics proteins. Metabolomics means protein and results of some proteins. Lipidomics, epigenomics, and interactomics, which is very difficult to understand. It has a branch of science such as systemic biology is this one, and many X already we have. I want to explain one basic part of genetic. The basic part is when we speak about genotype and phenotype. What's the difference between phenotype and genotypes? When we say genotype means DNA, RNA, and something which is written in the DNA. DNA has got a lot of information inside. Phenotype is what you see. This feature of me is my phenotype. You is your phenotype, which is coming from genome or genetic. So, to change phenotype, you have to change genotype. What this means manipulation of genotype. If you can change genotype, you can change the phenotype. Understand? If you change, for example, your eye, your hair, your height, your whatever is your phenotype, you can change it according to the genotype. The best way to know genome is the next generation sequencing, which I said to you, and the system which can change the genome is CRISPR, which I will explain tomorrow. Nature and Science, two very famous magazines, which explain about human genome and human genome. So human genome, which is this one, is very important. That's human genome. Genome is different from genes. You must understand. When we are speaking about human genome project started long time ago to know about, to discover A, T, C, G of human. Not each human, whole human. I mean one person or two persons, check all 3.3 multiple of 2 to 10 to 9 of sequence of human. Then check this one. But nowadays, now we can check human genome. This is the genome. Genome is different from gene. Genome. That's human genome from the first nucleotide to the end nucleotide 3.3 10 to 10. I can check using NGS. I can sequence all of your nucleotide easily now, not at that time when Human Genome Project started. We are speaking about genome. My question is it the same genome in all your cells or not? The genome is the same in all your cells or not?
Yes.
You have cell in your foot, you have cell in your hand, you have cell in your eye, your tummy, everywhere you have cells.
Yeah, our genome is the same.
The genome is the same. What is different? Phenotype is different. Why?
Which part is expressed as different?
Because the genes are different. Look, genes, RNA and proteins are different. Genome, DNA, is the same. But genome transcript, make a lot of transcript, means RNA, transcribe, and then translate to proteins. These two are different, but this is the same. Understood? So, the genes, when we say genes, means RNAs and protein. When we say genome, means DNA. Is it clear now? Understood? Okay. So, how many genes we have? 2,000, 25,000 to 30,000, something like that. Look at that. Our genes is the same as plant gene, the same as mouse gene. But am I look like mouse? Me? I don't think so. I'm not a mouse. This is a mouse, not me. So, mouse, I am sure I'm not a mouse. I am sure I'm not the Drosophila. So, the genome of the Drosophila is very close to us. For example, the gene and genome and thing. But why we are different? Because we have a lot of regulation of transcription. That's it. Regulation of transcription. Okay. It was just introduction from your high school. Now I'm going to speak about personalized medicine, which is this session today. Personalized medicine is a medical model that customizes. This is very important. We customize healthcare and decision and treatment to individuals. Each person. It means for each person we have different treatment, different because of the effectiveness of that. Human Genome Project start about 19, I suppose 1901, something like that, but finished 2003. It means we know about 10 years ago, something 15 years ago, 10 years ago, NGS as an instrument used for sequencing. And after that, we have a lot of sequencing data and so on and so on. That's development of sequencing. When I'm saying sequencing means to understand about 3.3 nucleotide. It means sequencing. Sequencing is a technology to understand, to determine order of nucleotide in entire our body, our cells. Okay, entire genome. What is the NGS in personalized medicine? You know NGS, I explained NGS for you. NGS is very important for profiling of genome. It means to know about the data, pharmacogenetics or pharmacogenomics, both, cancer genomics, which I will explain, non-invasive perinatal testing, NIPT, I explained before, rare disease diagnosis, importance of NGS in personalized medicine. In genetic and genomics, you remember I said genetics and genomics, genome and genes. It is very important, and also personalized medicine is very important for treatment and develop, provide the best possible care and treatment for each person, each patient. A few days ago, I asked ChatGPT, exactly this one. I asked ChatGPT, and I said, I am professor of the university. I want to explain about NGS and personalized medicine for medical students. What do you suggest? If I say to you, you say, no, I don't accept. But if ChatGPT say to you, oh, ChatGPT, okay, you accept quickly. That's AI, not modernizing I. So that's AI, ChatGPT. So what ChatGPT said basic genetics and genomics, NGS technology, and clinical application, some ethical which we have to be aware for. What I am saying about the results of NGS, a lot of data which we have from NGS, and medicine, your job. You are a medical doctor. You will be a medical doctor. As I said to you, in future, without NGS, you can do nothing. Definitely, you will see in future. So that's NGS. And I didn't say it. ChatGPT said. If I say, you say, okay, no. But when ChatGPT said, okay, yes, possible. Look, it is from ChatGPT. In future, you have dependency to NGS. Means dependency to whole genome. Okay, it was just introduction about personalized medicine, NGS, sequence, and so on. Now I want to explain more about that. I said we have 3.3 multiplied 10 to 9 sequence. How much is difference between two persons? You and you. No, you and your sister. How much? And you and your brother. You don't have brother.
No, I have.
You have. How much is the difference between your genome and your brother's genome?
So much different. Lot of different.
You and How much is the difference?
0.1.
Yeah. 99.9%, we are the same. We are the same. Only 0.1 difference. And we can see a lot of phenotype difference. I am not look like you are not look like another. In the world, in the whole world, whole different countries, you cannot find one person exactly the same as another, except identical twins. Forget about identical twins. Except identical twins, you cannot find two persons the same. Only with 0.1% difference. Only with 1% difference. 0.1% difference. Look at that. It's variation which cause differences, phenotype differences. A lot of differences between individuals is only this one. And today I am going to explain as a personal medicine your job. If everybody was the same, you are not medical doctor. Because we don't need medical doctor because we are the same. Patient the same, the pain the same, disease the same, if we are the same. But with 0.1% make job for you. Make job for you and me, both. It's our job. Understand? Understand? It is only this one. What are these differences? What are the differences between nucleotide? Just one nucleotide. Is it possible? Yes. One nucleotide in one gene is different with him, between us. Or maybe variable tandem repeats. Do you remember tandem repeats? Tandem repeats. Yeah. Do you remember? I said about 8. The paternity test, forensic medicine, and so on. Do you remember that one is variable? So we have differences. And also maybe copy number of the gene, deletion of the gene, and difference duplication of a gene, and some epigenetic variation which Dr. Prader will explain for you. Is exactly this one. Look at that. Some nucleotide. Do you remember that tandem repeats? Deletions, insertions, there are difference between us. It is happening like this. One nucleotide which is SNV or SNP, something like that. One nucleotide, one insertion, one deletion, one nucleotide, and so on and so on. That makes difference between us. Make differences between us. Look at that. That's the results of phenotype. These two dots, they have the same genotype. How much difference between phenotype? Huge! And also in humans. In humans is the same. Huge difference between me and maybe, I don't know, Bill Gates, and you, and Elon Musk. Maybe the same, I don't know. You are the same? Okay, he is the same with Elon Musk. So it is just joking, kidding. But in general, huge differences. Some of these differences, nucleotides, make harmless, some of them harmful, What means harmful? Have you seen? Have you? Yes, maybe. Have you seen in a family they have a lot of cancers? Yes, but some not. Another family doesn't have cancer. In one family they have a lot of heart attack. Have you seen? Many persons die by heart attack. They die. A lot of heart attack or a lot of cancers. And why? Because they have the same mutation. They have the same mutation which is different from another family. So that's variation. In this family, not in another family. This variation is just 0.1% of whole gene. Understand? Just only maybe one variation. Somebody has got C, somebody has got T, somebody has got G, and so on and so on. It's just one variation, but makes a huge difference between phenotypes. What is this called? Polymorphism. What's morphism? Morph coming from form, coming from shape, So we call poly means different, morph or shape, morphism. So one nucleotide make polymorphism, different shape. Somebody has got black hair, another one has got blonde hair. Is it possible? Yes, it is. It is possible. So black hair or maybe blonde hair. If, look at this one, if we have
You can answer.
no, take it off. If we have only 1% in the population, for example, we have two things. Mutation for thalassemia is mutation cause disease. Am I right? In a gene of hemoglobin. Thalassemia, which has got mutation in hemoglobin, means cause disease. What about one mutation which make blonde hair and black hair? Is it polymorphism or it is mutation cause disease? Black hair and blonde hair is disease? No, it is. Why? Because in population, this one is more than 1%. Population of the world, not population of Iran, not population of some countries, population of the world. However, in the population of the world, thalassemic mutation is not common. It's not more than 1%. Understood? It is just a standard. What are use of these 0.1% differences between our gene? What's that useful for? Can you give me some application for this one? Applications for this difference between different humans. Application. One of the application I said, paternity test. Another one,
Forensic medicine.
forensic medicine.
Determining the race.
Sorry?
Determining the race.
Yes. Others.
If someone has a disease trait, we can use this to
Maybe
find out.
related to disorder, related to disease. Association study. Association. This marker, look at this, For example, I have a gene, that's the DNA, DNA. This is a marker, and this is a gene. That's gene. And this is a marker, means marker. What is this marker? Is SNP or STR. Repeats. If this marker is close to this one, you can say, okay, I got this gene. This gene is, for example, this is blonde hair. Or this is a disorder gene, disease or disorder. If I get this marker, which is close to this one, if I get this marker from my dad, my father, means I got this gene from my father. If I have, for example, during pregnancy, you want have a blonde hair baby. You remember PGD?
Mm-hmm.
You want a baby, very good baby, blonde hair, and a blue eye. Yes, maybe. Father has got blue eye and blonde. Mother against black and brown, for example. Which one do you want? You come to me, say, which one do you want? Mom or dad? Give me the money, I do that for you. To be honest with you. Well, how? I don't check the gene. I check the marker close the gene. Why? Because if this gene is blonde hair, it's coming from mother or father, and I check and find which one is from mother, which one is from father. This marker is, for example, A marker, father has got D marker, S marker, something like that. For example, I check which marker coming from your dad. Do you remember the amount of repeats?
Mm-hmm.
Father has got 8 repeats. Mother has got 6 repeats. This one is blue, in this one is black. This one is blue. How can I check the baby? Which one do you want? Blue, black? Which one? Blue? Blue. Okay, blue. This one. Blue one, so which one I have to choose in the tube? 6 months. Understood? Understood the job? How? Because it is different between people. It is that 0.1%. You understand what I mean? Is it useful or not? It is personalized. I said about blue and white. Is it possible it is a disease? It is a deafness? Yes. Deafness and healthy without deafness. Understand? So we can do. Another thing which we can do that is ancestry tracking. Do you know ancestry tracking? What is that?
For example, there's a company, I don't remember the name exactly, but something.
From US, 23andMe.
Yeah, 23andMe, and they track their roots. For example, some people have Caucasian roots or South African.
Where is coming from?
Yeah.
Where is your origin?
Exactly.
Yes, exactly, ancestor tracking. It's ancestor tracking. Where is it coming? That's from Muhammad, one of my friends. And we can find the ancestor tracking. For example, 97 is Iranian. 97 is Iranian. 0.05% is Egyptian Arab, and so on and so on. 1% is Asian, and so on. 0.2% Indian. That's from ancestry. You can find the way, where it's coming from. For example, according to the haplotype means marker. You remember the marker? I can check in different people. And the marker, oh, it's coming to this way, this way, this way, and so on. That's for people. Or for different people, you can see this go to this way, and that way. This one is myself. This one is for myself. Because usually when I design a test, I do for myself first. And we do array, a SNP array, you know. So I check first for myself, where am I from?
How much does it cost here in Iran?
Me?
Yeah.
Where I'm,
Yeah, where are you from and how much does it
I can show you. Don't worry. Because it is not, I cannot show for my patients or my other people because it is not ethical. But I can show myself. It's not ethical. That's why I can say it is me. Don't worry. So you can see here, when I check, I am in this area. Yes, I know it is in this area. 69 Caucasian. I have to read here. I don't have my glasses. 8% from Syria, Arab area. I know because I am saying my origin is from Saudi Arabia, possibly. So 22% from Pakistan and Afghanistan, maybe, and Asian, 22% Asian and so on. This area is my origin. When I check this marker, look at this marker, exactly this marker, not this one, sort of marker. When you check this marker, I can say during about 20,000 years, during 20,000 years, my ancestry, my father and father, is it father or mother? I mean, mother. My coming from Europe, it is right because my grandma was blonde, and usually I said, "Oh, you were some Iranian captured you from Russia or some from Rome, Italy, and took you here at that time as Chinese." You didn't know that. So usually I said to my grandma, just kidding, because he was completely blonde, and I used to say, okay. So when I check this one, I say, okay, maybe my origin coming from Europe. And when we check, we say this is similar to these two persons, 20,000 years ago. For my father, we check these one, about 42,000 years ago. I mean, during this period of time, we are very close to these persons. It's not, we are very close to this person. I mean, in our origin. Do you know what's the similarity between us and these persons?
On the left one, the same color here.
That's weird. He is Jewish. He's Jewish. He's Jewish.
That's the color.
And I am saying, no, I'm saying our ancestor is coming from the same Abraham. The same ancestry. Understand? Yes. Because you're Jewish.
So you have Jewish ancestry.
Of course, I am saying I am coming from Muhammad and Abraham and so on. So, should be the same. Should be. I don't know, but many changes. So, we can check the ancestry. That's what I am saying, what I am explaining now to you. It's about using of 0.1%, 0.1% for tracking ancestry. For example, not only this one, I want to explain many things to you. I mean, sometimes, look at this word, sometimes, it is very important word to you. You have to memorize this one. Be careful and carefully listen to this one. Sometimes this marker doesn't do anything. It is. Do you know how many percent of our genome change to genes? Do you know? No? You don't know?
0.1%. 0.1.
No. From genome, how many percent of this genome change to genes? How many percent of this genome? How many percent of this genome change to RNA? DNA, RNA, protein function. How many percent of this genome change to RNA
10%.
and protein and function?
10%.
No.
All of it?
No, not at all. No.
Close.
Who said 10?
Me.
No. Go less.
5.
1.
5.
Less than.
Between 5 to 3%. Only 5 to 3% change to genes. Others are nothing, maybe. We don't know at least. We don't know at least. So we say junk DNA. However, they are not junk. You know junk? No junk? No. We say junk, but however, it is not junk. Is it clear?
Yeah.
So we say junk. Now, what I am saying now, I want to say sometimes this repeats. This STR is not here. It is here. Do you know? If it is here in the gene, it means translate to protein. It means maybe our protein is different. You understand what I mean? Means your protein is different, my protein. I am not saying about ancestry and tracking and something, or forensic medicine and so on and so on. I am speaking about translation of DNA, transcript to RNA, protein, and function. Your protein is different from my protein. Is it possible your protein has got different function from my protein? Yes. Yes, a lot. Yes, of course. So sometimes the protein is in the gene, means in the here, because you know exons, introns, and splicing and changing to RNA, mRNA, and mRNA to protein. You know it. This one. Okay. I want to give you some example of this one. When the protein change, the function change, the protein change, the results of that is you are, as a medical doctor, you have to have different approaches for me and him and you and others, because our protein is different, our function is different. Is it clear? Is it clear? Okay. I want to give you one example, which is very important example. Do you know thrombophilia? Do you know hemophilia?
Yes.
Hemophilia, what's the meaning of hemophilia? Means bleeding cannot stop. What about thrombophilia?
Excess of thrombocytes.
Lots of clotting, lots of blood clots.
Yes, means
Blood clots quickly.
clotting. Clotting is high. Clotting. Thrombus is high. Clotting is high. It means thrombophilia. Philia means like. Thrombo means thrombus. Thrombus is clot. Philia. So we have a factor 5 in our body. Do you know the system of factor 1, 2, 3 in clotting system? Okay. Factor 5. There is a mutation or polymorphism in factor 5 called Leiden. Very famous mutation. When you have this Leiden, I want to give you an example which is very important for you, especially for you, more than them, but it is very important. Let me give you an example. A woman is going to travel from here, where we are? Here, okay. From here to Toronto, not Toronto, let me go other side, to Vancouver. Vancouver. You know Vancouver?
Yes.
Okay. From here to Vancouver. Okay, we have to go to Dubai, and from Dubai to Vancouver. How long does it take? 11 hours. 12 hours, something like that. 12 hours traveling. Maybe you are not rich. Maybe you are rich, go to this business. Maybe not rich, you go to economic. And sit like this for how long?
12 hours.
12 hours. There your blood is. The blood.
Legs.
Blood is in your
Legs.
Yes, for 12 hours. Doing like this. You cannot do just say from 12 hours like this. Just collection of your blood is here. Possibility for clot here. Okay, doesn't matter. For me, doesn't matter. For you, maybe doesn't matter. If you have mutation in factor 5, possibility for this one is comparing to normal one with one mutation. That's the DNA. You have 2 alleles. If you have 1 allele, it's 10 times. If you have 2, is 40 times possibility for thrombus, for clot. If you have contraceptive, do you know contraceptive?
Yes.
Contraceptive. If you have contraceptive, possibility for clotting is 400 times. It means after traveling, somebody eat contraceptive and has got 2 mutations here, and traveling for a long time in economy. When you got Vancouver, they have to call emergency because you have clot here, and this clot go to
Heart.
Hey.
Brain.
brain. you have a stroke. Understand? They have to take you out, not horizontally, completely bed rest. So the result of that maybe it is dead. Understand? Is it important to know?
Yes.
Is it important to know? Do you understand personalized medicine? Okay, you yourself now understand personalized medicine. That's why I am saying when I design a test in Iran, always I check out the first myself. Fortunately, I don't have this one. Another one is MTHFR, methyl tetrahydroxyflavin. Is it another one? This one is in Iran, is very popular. I mean, this polymorphism is this one. This is also made thrombophilia. Do you know another possibility for thrombophilia is not deep vein thrombosis? It is, yes, make miscarriage. Do you know miscarriage? Miscarriage. Make miscarriage. So, make miscarriage. Do you know why? Because there is a lot of blood vessels in chorion. And chorion become, I would say, comparing to young chorion, it's made old chorion, older than normal, comparing, veins of chorion, or bloods, artery in chorion. Another polymorphism, what I am speaking about polymorphism which can make function. Another polymorphism is lactose intolerance. Do you know lactose intolerance?
Yes.
When you drink milk, you go to toilet. Very clear. People know sometimes. No, you don't have it if you don't go. If you don't go, you don't have it. Do you know in which countries is very, very popular?
United States.
China. Do you know in which country is not popular? Frequency is very low. USA, Russia, Australia. Understood? In Iran and this Middle East area is between. It is very, very interesting. When you go to, not now, long time ago when I used to go to China many times, when you go to China, usually you cannot find yogurt, milk, and so on, popular in supermarkets, popular, long time. Now it is popular, but they don't drink. They don't drink milk, they don't drink yogurt, eat yogurt and so on. However, we do. And it is very popular in, understand why? Because when you eat, go to toilet. Understand? Why? Because of the polymorphism. Why? Because of the gene. Because of this one. Is it clear?
But doctor, I always drink in the milk.
Sorry?
I'm always drink milk, but there is nothing between.
Doesn't happen.
Normal.
No, because it is not popular in this area. It's not, because you don't have the problem. You don't have that polymorphism. That you don't have what somebody has. Understand?
Yeah.
How we can check it? The sequence by NGS, by array, by checking the sequence. It is possible. You can check it. You can check it. And we can check if lactose intolerance or not. Not only for lactose intolerance, for many things, many allergies, asthma, or fever, or dog allergy. Do you have allergy to dog or not? Many different allergy. It is possible to check. You have
From Sajjad I have allergy.
allergy to dog?
No,
it's
from Sajjad.
Okay, it is. How we can check it? By polymorphisms. We can check many allergy. Understood? That's allergy reports. Human polymorphism, different polymorphism. What I am saying now, I am giving you some examples in different polymorphism in our body and make different things. For example, polymorphism for G6PD. You are a student and you are going to medical doctor. G6PD is common in this Middle East. Do you know what's that?
Yes.
Holocaust.
Pharmacogenomics.
Phobis, exactly. Phobis is you prescribe for a person who suffers from G6PD deficiency or phobis. Do you know what happened? You kill him or her. You kill him. You kill him. You prescribe aspirin. You prescribe, for example, vitamin K. You prescribe, for example, ascorbic acid, vitamin C. You kill him. Do you understand what I mean? Is it important to know the polymorphism for people or not? Do you remember? Not me. ChatGPT said to you, not me. ChatGPT said to you, in future you cannot do treatment, medicine, medical job without sequencing information. Not me. I didn't say. When I say, oh, look at what else said. No. ChatGPT said. AI said. Make some credit for that. Now you can understand. Do you know HLA typing for transfusion?
Yes.
Transparent colors. HLA typing is based on polymorphism. Different HLA typing for transplantation. Without HLA typing, no transplantation. Because different people, they have different HLA typing. Exactly yesterday night, I was in taxi here in Kiev, just speaking with a phone with somebody else, which I don't know the driver, said, unfortunately, the HLA typing for the person didn't work or something like that. You know for what? Leukemia. Transplantation for leukemia. Between two brothers. It didn't work. Just by chance yesterday night I was just hearing from the driver. HLA typing because of many polymorphism on protein. It is one of that. Many here. Tandem repeats. You remember tandem repeats? Tandem repeats. It can be 3 or more. I want to give you an example for tandem repeats. You know what? Which one? Do you know what is this? Do you know androgen?
Yes.
What's androgen?
Yeah, hormone.
Hormone.
Like,
Do you know androgen?
Yeah, that's true.
Body building. Androgen. They It's androgen. Bodybuilding, androgen. It's testosterone. Is the male hormone. Is it right? Androgen has got receptor on each cell. Androgen, receptor of the androgen. Androgen is not protein, but receptor is protein. Is it possible this androgen has got a repeat? Do you remember the repeat? Sometimes some person has got 5 repeat, another one has 8 repeat. What happened? Repeat is here. Look at that. It's 5 repeat. This one is 5 repeat. This one is 8 repeat. Look, 5, 8, and this is androgen. For this one, match androgen with receptor easier, or this one easier? Which one? First one. 5 is difficult because it is like that. It's difficult to. This is androgen. It wants to stick androgen, stick to receptor. This is 5, and this is 8. Which one is better?
8.
Which one has got more function? 8 or 5?
8.
I didn't say, you say. Is it clear?
Yes.
So repeat. What's the function of androgen? Do you know many functions? Many functions. From baldness, hair, to infertility, cancer of prostate, breast cancer, from infertility to cancer. A range of function for androgen based on a repeat. Understand? Is it important to know this repeat for your job in future, for your job? I mean, when you graduated?
Yeah. Yes.
I want to give you one example, one very important point. Have you done lab tests for yourself?
Yes.
Done? Have you checked, especially for female, have you checked your hormones?
Yes.
Many times. What do you check? Hormone or receptor? Hormones in your blood. Never ever check receptors. Never ever so far you check receptors. Because for checking receptor you need NGS. Understand? Understand what I am saying? In future, you don't check FSH, LH, testosterone, estrogen, progesterone, and so on in the blood because it is useless. You have to check also receptor. Understand? Is it important in future or not? Is it clear? That's what most of the receptors, they have repeats. Understand? Is it important to check? I don't say, you say. Next time I ask ChatGPT to give you. Okay. I said some example of repeats for your medical jobs. I want to give you some example about some polymorphism in lifestyle. Do you know lifestyle? Very well. Very. Psychology. Is it lifestyle? Psychology.
Yes.
Psychology report. Which kind of report? Alzheimer disease. Different sort of disorders. Anger. Do you know anger?
Yeah.
Anger. Do you know, for example, depression?
Yes.
Yes. Is it important to know about our depression, our anger, our many things for our life?
Yes.
I suppose yes. And how we can check it? Look at this. Many polymorphism we can check. And we say, you are in high anger. Is it this person useful to be a medical doctor? Yes or no? High anger. Look at anger.
No.
Look. Very aggressive. No. No. Is it useful, for example, when you want to pick up a person as a soldier, to know about this one? Yes.
Yeah.
It is very important to know about psychology, about bipolar, about schizophrenia. It's a media. For example, depression, I said, anorexia, or other things. What about the sport? Which one is a sportman or a sportwoman here? You? And you? No, not a sport. Do you like to know about sports? Me, I am thalassemia minor. Me, myself, I know. I know thalassemia minor. So I cannot go for be as a sportsman because my hemoglobin is lower than you. So I cannot be a good sportsman. However, I can live. Nothing happened. 80 years possibly. And so, is it important to know, for example, about that one? Ability, athletic ability. Is it good to know? Do you, in this country, recently check before that time? Now, is it important to know if you want to go for athletic, to be an athletic, high-ranking athletic, to check your ability? Of course.
Yes.
Very important to know that one, sport performance. Which one your ability, for example, you are in the last one, for example, body fat mass. If your body fat mass is less, it's good. Or something about VO2 mass and others, any different parameters about sport, any different parameters about skiing. I'm sure you like that one. Women like that one, skiing. Maybe. I'm sure, 100%. Skin pigmentation. Do you like to know? Of course. What I am speaking about? Lifestyle. Do you understand lifestyle? Polymorphism and lifestyle. Do you like, for example, facial wrinkles? You don't know? You don't want to know? Definitely you want to know. I'm 100% you know. Because you buy a lot of something you use for your face. Okay, facial wrinkles, I'm sure you want to know. Many others we check already. We can check many parameters on skin according to, look at these genes, many genes, polymorphism. Understand? Is it useful for lifestyle? Of course yes. Of course yes. What about nutrition? Is it good to know about? We eat a lot or not? For this one, I can show you something about myself. This has happened to me, not others, but this one happened to me. Look at this one. I check this one for myself. long time ago, I used to, usually when I use brushing, I had bleeding many times. So I was thinking maybe I suffer from leukemia because I had a lot of bleeding. I said, oh, my blockade is down, and so I suffer from leukemia. When I did this test, this one is mine. I did this test. Look at that. Which vitamin is very, very low?
Vitamin C should be.
Vitamin C.
Yes, absorption of vitamin C in my digestive system is very low. Yeah, vitamin C. Vitamin E is okay. Vitamin A is middle. Vitamin D is good, not bad. But C is low. Do you know what's the cause of bleeding?
Scurvy.
Vitamin C. That's why every day morning I get a pill of vitamin C. And now I'm absolutely better. Understand? Is it useful for our life? Definitely. Definitely. Talent. Do you know talent?
Yes.
Is it good talent report? Yes, of course talent report is good. Because, for example, we can say, okay, don't go for medicine. Go for engineering. Because for example, your mathematics is much better than your memorizing. We do that. Now, we say to people, is it good for your lifestyle?
Yes.
According to what? According to polymorphism. According to polymorphism, we say, okay, you have talent for the music. You are good musician. You are not good musician. Is it useful? According what? According that polymorphism. Is it useful? Is it possible to say about health condition? You know health condition? Yes, it is. About arterial hypertension? Do you know about myocardial infarction? I like to know about possibility of my myocardial infarction is high in my body or low. Because according to that one, I can change my lifestyle. You understand? I can change my food. I can change my sport. Go to a sport every day. I can change my life. So it is useful. Diabetes. It's very good to know about the diabetes. I am, for example, hearing diabetes, or here. Which part? It's very important to know according to these polymorphism. Other conditions, many conditions of health you can see. So, what I am saying now about personalized medicine. Personalized medicine in medicine, treatment, or prevention and lifestyle. Understand? That's personal. According to the differences, where is that? Our differences in our gene. Is it clear what I'm saying? Is it useful? AI or ChatGPT said, do you agree with ChatGPT? Okay. Good. Good. Personalized medicine is this one. And also, how many pills you can use in personalized medicine. Okay. What I said something about personalized medicine. Afternoon, some left. Afternoon, I give you some others about personalized medicine and drugs. How much drug you can use and how much drug you have to prescribe, which drug you have to prescribe, more or less, according to personalized medicine and according to genetics. Genetics means this one. According to this one. I give you after. And another session which is about malformation. I suppose we have to have